Thank you. Okay. Thank you.
So, we're looking at a blood smear here.
Oh,
The majority of the cells are what kind of cells?
red blood cells?
And what are we going to call this cell right here? Yeah, neutrophil. Remember, it's got a nucleus. The nucleus is segmented or like a string of sausages, right? That is folded on top of one cell. There aren't really any staining granules in the cytoplasm. So we'll go with neutrophil. What about all these little speckles here in the clump? Good. So, higher power, same song, second verse, neutrophils, right? Segmented nucleus, nothing happening stain-wise in granules in the cytoplasm. What about this guy? Eosinophil, all right? So you can just see the nucleus. Notice there are a couple of granules sitting on top of the nucleus. And those nucleus are clearly eosinophilic or pink stain. The granules, excuse me, are eosinophilic. Basophil, okay? Not a lot of granules, but they're definitely there, and they're purple. They're not that eosin color, all right? So, I guess with respect to identifying blood cells, you can expect a micrograph, and you can expect it to be clear, a clear micrograph. I mean, some of you have looked at some of your blood smears. There's some things there that are equivocal. Can't really say what it is, all right? You're not going to see that on the exam. You're not going to see equivocal things on the exam. What about this one right here? Right. Two things. Look at the size. Much, much bigger than the red blood cells. Large nucleus. It's kind of got that kidney shaped or indentation. Lots of cytoplasm. Lymphocytes. All right. All right. So that was a quick move through the blood cells. Let's look at muscle tissue here. Again, it's important to keep a sense of scale here. As Nigel mentioned, these cells can be as long as your upper limb. All right? Their width is about 100 microns. Cardiac muscle, maximum length really is 100 microns, maybe 20 microns thick. Smooth muscle, maximum length 200 microns, but only 5 microns thick. So there's a big difference there. All right? So clearly we've got striations. We can see that one cellular border is here. We see, looks like nuclei, although some of this could be capillary, nuclei associated with a little blood vessel. One cell here, another here, here, here. Most of the nuclei look like they're peripherally oriented, right? You have a few, like this guy right here, which is probably sitting on top of the cell. So you're looking down on top of it. But again, you have to keep that in mind. That wouldn't be what I would call a centrally located nucleus. It would be one that's on the periphery, but you're looking down on top of it. Clear striations there. Higher power shot. Maybe you can see the border of this cell here. And now we see these nice nuclei, multinucleate, peripherally oriented, with striations. If we look in cross-section, we don't have the striations, but we've got these polygonal-shaped cells, and again, all of the nuclei are peripherally oriented. So that you're telling yourself pretty much got to be a skeletal or striated, visceral striated muscle. This is a nice shot here, a Gamore's trichrome, so a skeletal muscle. So here's your skeletal muscle fiber. You can see the nuclei kind of have that red here. Then you've got this little capsule-bound structure with smaller profiles that look similar to that. These are actually intrafusal muscle fibers. This is part of a Golgi tendon or a muscle spindle, a muscle spindle, which helps your body sense change and rate of change of your muscles in terms of shortening. So if you see that, count yourself lucky. That right there is probably a nerve. Again, this is a frozen section, and you're picking up mostly Schwann cells of this nerve coming around here. Another frozen section. You can see endomesial connective tissue in among these myofibers. Here's another nerve. See the red here? This is a stain for cholinesterase, which is an enzyme that degrades the acetylcholine over the neuromuscular junction. So what you can say is that these are sites of neuromuscular junctions. It's kind of an immunostain allowing you to see that. What are we looking at here?
Cardiac.
Okay, how do you know it's cardiac muscle? A little louder. Right, you've got centrally located nuclei. There are a bunch of clues here. If we had a fine focus, we could probably, and turn our aperture diaphragm down, we could pick up the striations. A little difficult to see here. The other thing, as Garrett mentioned, you've got these central nuclei. And notice that this nucleus, it has that halo or that clear zone around it. That's because the glycogen that was there in real life has been washed out. That will be useful, seeing that halo in cross-section when you don't have striations, helps you tell yourself you're looking at cardiac muscle. The other thing that's really cool about this image is, look at that structure there, here. This one looks like a staircase here. Those are your, exactly, intercalated discs. Don't freak out if a glass slide, we tell you should be able to see intercalated discs. You don't. Just look at more cardiac muscle. I mean, you have to really play it fine to see them. It's there. Fortunately, single centrally located nucleus with a halo and striations get you there anyway. All right. Keep in mind that the heart muscle is surrounding chambers that are spherical or spheroidal, right? So the muscle in the walls wraps every which way. So in an individual section, you're likely to get sections that are cross-section to the long axis of the cardiac myocyte, oblique sections, or long sections. So look around through the whole slide and you'll be able to pick out any one of these orientations almost without fail. All right, what are we looking at here? Low power, I will admit, but there are enough clues here. Well, one thing I'd want to be thinking about is what this is right here. And the other thing, if you look here, like right in here, you see that little dark speck with the halo? That's a cardiac myocyte cutting cross-section through the nucleus. Where did that go? Right? Look right in there. All right, so even at low power, there are clues. James, you look like you're frowning. You don't see that? You see that nucleus? You guys can't see that stuff?
No. Is that adipose tissue up in the left?
Yeah, definitely.
Okay.
I can't help you if you can't see it. I would suggest that you keep in mind central nucleus with that glycogen filled, what becomes a halo when you wash it out, will be helpful. So, I got no answers about what that is. Or I did and didn't hear them. Purkinje, right. I think I've got a higher power. And we've got an example of a binucleate Purkinje fiber. First, look at the size of these cells relative to a cardiac myocyte down here. They're big. If you look carefully with high power, you might see the remnants of the contractile apparatus, all right? Usually that's downplayed and glycogen synthesis is upplayed. So when you fix it, you end up washing out, and that's why they look pale. This is a single smooth muscle fiber that's been teased out. So again, remember I said they can get up to 200 microns long, but at their thickest, right here where the nucleus is, a little less than the diameter of a red blood cell, okay? So if you think about these cells that are tapered very, very thin like that, you can imagine how they look when they're packed and cut in cross-section. So let's look up on the top. I mean, I would guess, but I'd have to see a lower power orientation, that this is circular smooth muscle of the muscularis externa, and out here we caught the outer longitudinal muscle in cross-section. So if you look at this, you can see that there are all sorts of different diameter circular profiles, as well as some nuclear profiles. And you see the different diameters because you've cut that tapered muscle cell, which is packed at different levels with reference to the nucleus. So you're going to see a smaller circular profile the closer you are to the end, bigger as you get closer to the nucleus. And if you spend a little time, starting to die. That's probably close to a nucleus, getting a little smaller, getting much smaller here, and clearly you can see the circular profiles where we've cut it through the nucleus. So, here's Auerbach's plexus, a little bit here. Notice that the neurons, cytoplasm kind of is a purplish color. That's something that really helps. So, Auerbach's plexus, gut, you want to look at the interface between the inner circular and the outer longitudinal. And if you do that, circumnavigate yourself around, you'll find areas like this, which represent myenteric plexus of Auerbach. All right. Nervous system. I don't have a slide to show you of the brain. Keep in mind that the concept that we want you to get there is that the gray matter is on the outside, white matter runs in tracks on the inside. When you do neurology, you'll be dealing with it in a lot more detail. All right? When we get to the spinal cord, the arrangement of gray and white matter reverses. All right? As Nigel mentioned. So here we have our butterfly, which would represent white matter,
White matter.
gray matter, and surrounding, the dorsal, ventral, and lateral tracts, which is all white matter. Sensory input is what? Through the bottom here? That's motor output through the bottom. Sensory input through the dorsal horns. So again, basic concepts is what we're looking for here. All right, this is a nice shot of a multifascicular nerve, process paraffin section H&E. Okay? So how many fascicles do we have here? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 or so. They are all held together by what kind of connective tissue? Epineurium. Good. Which is a dense, irregular connective tissue. The connective tissue that defines each fascicle we call perineurium. And then the endoneurium is the connective tissue that runs in and amongst the myelinated and unmyelinated fibers within each fascicle. All right? With the paraffin processing, we're removing lipids, right? Think about adipose tissue that we looked at. That big old central vacuole, it's gone. It's just a clear space, okay? So the myelin sheaths here, which are about 80% lipid, most of them have been dissolved away, so we don't see it. If we use osmium tetroxide, which actually fixes the lipids, they remain in place. And so all these little dark specks, little donuts would represent cross-sections through axons surrounded by a myelin sheath. All right? And out here, just see at the corner adipocytes that have that fat in place in that fat vacuole. All right, here's an electron micrograph. Most of what we're looking at here, with the exception of this fiber and this fiber, are unmyelinated fibers. So, in reality, this is a very small myelinated fibers. Remember Nigel mentioned they range from about 1 micron to 15 microns. So we're down at the lower end here. But that's clearly a myelinated fiber. And in this section, we have the nucleus of that Schwann cell that form that myelin sheath. Okay? If we section above or below the plane of the screen there, we might lose contact with that nucleus, but we'd still have the myelin sheath. All right? Here's an even smaller myelinated fiber right here, axon, just maybe 4, 5, 6 lamellae, or wraps of the myelin sheath. I think that's all she wrote. So, have a good weekend.